Let's go ahead and talk about meiosis. And meiosis is simply a modification of mitosis. And because meiosis is a little bit more involved, we're going to break it up into two phases. Meiosis 1 and Meiosis 2. And then we're going to take each phase of meiosis, Meiosis 1 for example, and we're going to break it up into four more phases. Prophase 1, Metaphase 1, Anaphase 1, and Telophase 1. So let's go ahead and start with Prophase 1. Now a lot is going to happen in Prophase 1. So I've got an early Prophase 1 and a late Prophase 1. And let's take a look at the chromosomes here. I've shown 6 chromosomes. In a human cell you'd actually have 46, but for simplicity I'm going to draw 6 chromosomes. And let's assume that out of those 6, 3 originally came from Dad and 3 originally came from Mom. And these chromosomes, I've shown them, they actually have a centromere here. They've actually replicated already because remember, before Meiosis 1, we've gone through G1, we've gone through S, and we've gone through G2 of the cell cycle. So these chromosomes have already replicated. And because these chromosomes have replicated, they're going to be referred to as dyads because you've got one plus you've got the original plus the replicated one. And I'm going to fudge things just a little bit there because that's not quite the way it is, but for right now, that's the way we're going to depict it, as an original and a replicated. Dad's chromosomes, original ones, are going to be in blue, and Mom's are going to be in red. So in early prophase, the chromosomes are going to condense, the nuclear envelope is going to begin to break down, and the spindle apparatus is going to begin to form. In late prophase 1, those things have continued to occur, but in addition, homologous chromosomes have synapsed. That means similar chromosomes. Remember, you have 2 of each type. So those 2 of each sets are going to find their homologs and they're going to pair up. So I've drawn these in different sizes. Here's a small one of Dad, here's a small one of Mom's. And they're going to pair up. The medium ones are going to pair up. The large ones are going to pair up. So homologous chromosomes are going to synapse or pair up. They're going to find each other. And when they do, we're going to form a structure called a tetrad, because in a sense it's made up of 4 chromatids or 4 pieces of DNA. After that occurs, something interesting is going to occur. This is where we're going to increase genetic diversity. And remember, that's the point of meiosis, is to increase genetic diversity. And something called crossing over between homologous chromosomes is going to occur. So here we have a tetrad. And where the tetrads actually are, here we have a tetrad, and where the dyads actually touch each other, where the red touches the blue, those regions, here and here, usually there's about 15 on each tetrad, in and around 15. Those regions are going to be called chiasmata. And they're simply points of contact between the homologous chromosomes. And these points of contact, these chiasmata, is where crossing over is going to occur. Now here I have a model. Here's my tetrad right here. And as I've drawn on the board, the tetrad kind of looks like this. But in reality, that's not how they're paired up. They're actually paired up back to back, just like that. They're going to be paired up back to back. And wherever these DNA molecules actually touch each other, these points of contact, that's where crossing over is going to occur. So I'm going to go ahead and I'm going to I'm going to take 3 beads off of the red, and I'm going to take 3 beads off of the blue, and I'm going to swap them. And this is what we have here. This is where the 2 chromosomes have been, right here. This is where the swapping has occurred, the crossing over has occurred. And that's going to happen at about 15 points on average, give or take some, on each tetrad. And that's going to increase genetic diversity. So that's one place where genetic diversity is going to occur. Then from there, we're going to move on to Metaphase 1. With Metaphase 1, these tetrads are going to line up on the equatorial plate, right in the center of the cell, this region in the center of the cell. And then we're going to have our next place where genetic diversity is going to occur. And this is called random positioning of the tetrads. So here's one tetrad, and what I mean by that is this blue one has a 50-50 chance of being on the right or being on the left. And the same thing goes for this next tetrad. Here I showed the red on the right and the blue on the left, but again, there is a 50-50 chance that it could be just the opposite. The blue could have ended up on the right and the red could have ended up on the left. And the next one, 50-50 chance, 50-50 chance, 50-50 chance. So in a human, each one of those 23 tetrads would have a 50-50 chance of being on the right or being on the left. That's going to increase genetic diversity because when these chromosomes move to opposite poles, we're going to end up with, in this case, different colors on the opposite poles. In real life, we just have different chromosomes on, moving towards opposite poles. So that's the second point of increased diversity. The third place where we're going to have increase in diversity is going to be fertilization, but that's not going to be part of meiosis, but that is the third place where genetic diversity would occur. So, random positioning of tetrads. And we have a special name for this random positioning of tetrads. Mendel called this independent assortment. So these chromosomes, these tetrads, assort independently of each other. This one has a 50-50 chance of being here as opposed to here. And then when we move to the next tetrad, again, it's a 50-50 chance. This one is not influenced by that one. After metaphase 1, we're going to move on to anaphase. And just like mitosis, the chromosomes are going to break apart and they're going to move to opposite poles. All right? The centromeres are still intact, which actually holding these two dyads together is a protein complex called the synaptonemal complex. So that synaptonemal complex actually starts to break down the chromosomes, the dyads in this case, separate from each other and start to move towards opposite poles. Then we're going to start dividing the cell in two. So you can see our cleavage furrow beginning to form there. At this point, the nuclear envelopes begin to reform, the spindle apparatus begins to disappear, the chromosomes begin to relax or decondense, and cytokinesis begins to take place at the same point in time. Remember, cytokinesis is not actually a part of meiosis. It is a separate mechanism happening at the same time as meiosis. So that is also going on. At this point, we're going to end up with two cells, 1 and 2. And each of those two cells are now going to go through another round of meiosis called Meiosis Two. Let's go ahead and follow one of these cells. Let's go ahead and follow this cell right here through Meiosis Two and see what happens.